salmon are structured into multiple distinct breeding populations. So unless you have a very, very small river system, it's likely that you've got a number of distinct breeding populations in your system. But we just don't understand what these spatial boundaries are and what's defining the populations. So we were able to use that genetic information to look at the population origin of the samples and assign the numbers of fish that came from these different parts. And it was actually counterintuitive. The Euchre, which was the more distant river of those rivers there, was actually contributing most of the fish to that fishery. And as a consequence of that, they actually changed the way they managed that situation. In fact, I think the fishery is more or less shut down now. At the same time, we could use the genetic information to start to understand something about the breeding size of the population. So one of the things when you're a manager, the argument is, well, there's not many salmon that are actually there, there's not many breeding, we need to supplement it because the populations are small. Well, you can actually use the genetic information that you can collect by analyzing these markers to understand the numbers of breeders. And I won't go into the detail there, other than to just say, if you think about it, the more breeders you have, the more genetic diversity you have, given that there's individual variability. So by looking at the amount of genetic diversity and also the way it's distributed amongst individuals, you can start to draw conclusions about the numbers of breeders. This is potentially a very useful management tool. If you think about it, if you want to count numbers of breeders otherwise, that's an awful lot of field work that you've got to do. You've got to put traps in, electrofishing, etc., etc. And this is perhaps a much more useful management metric. At the same time, you can combine the information from these two here and potentially start to say things about levels of exploitation. If you know something about your numbers of breeders, and you know something about the numbers of fish from different populations that are being caught, you can start to say something about exploitation. So genetics is really just starting to open these doors. It's an incredibly useful tool. And the thing perhaps that Andy didn't dwell on too much was the fact that what we have here also is a hierarchy of genetic relatedness. And here's the Fiochan populations here, and all the ones from the Fiochan rivers, they cluster together genetically. They're more similar to each other. And then you go out to the neighboring rivers and you find here that again is that evolutionary development of these populations has led to them being more closely related based on geography. And again, you see it on the final scale of the whole northern Atlantic. These are Canadian American populations versus European populations, and you can see the divergence is greater. In this case, this divergence is associated with these having a different chromosomal number than these. They've evolved. They've been separated for in the order of 600,000 years. So our populations different in ways which increase the survival in rivers and marine areas they inhabit. Are they locally adapted? Well, Andy's talked about it in relation to trout. I'll give you some ideas in salmon. Now, this is a study. Phil McGinnity was a co-author, but Andy's also involved with this one. And this is, as far as I'm concerned, one of the seminal studies that have been done on Atlantic salmon and has an incredibly important management message. Now, what they did was they went to the Burish Hole system, which is here, where there's an experimental trap, an experimental station that's been there for many, many years. And they took one of the tributaries and they put a trap on it, and they introduced Burish Hole native fish and Owenmore fish, which are from the catchment here. That's the Owenmore River, so the two catchments back onto each other, but they're 70 kilometers from one mouth to the other by sea. And they monitored the relative performance of the fish at different life history stages. And the higher, this is 100%. If it's there, that means the fish are behaving equally. If it's higher, the introduced Owenmore fish are behaving better. If it's lower, they're performing worse. What you can see is that the performance of the Owenmore fish is worse than it is of the native Burish Hole fish, with one exception. They seem to leave the system much better than do the native fish. But the message here overall is, when you look at here, the overall fitness in relation to this of the introduced fish is 17%, or something in that order, of that of the native fish. Now, this is quite amazing when you think that's wild fish from one river put in before any hope of domestication occurring, and they've just been put straight in the stream, and you're getting that big difference in performance. And that's two catchments which share roughly the same kind of water quality and situation. And the biggest part of that difference is their ability to return to the native river. Now here's another one which is adaptation on a much larger scale. And Gyrodactylus salaris is a real scare story, particularly in Scotland. I put it down here as well, but perhaps to a lesser degree. But Gyrodactylus salaris is a parasite which is native and endemic to the Baltic region. And it's been introduced into Norway, and it's now basically caused the salmon stocks in up to 40 rivers to go extinct in Norway, and they've got a gene banking program, an eradication program, reintroduction of the native stocks, etc. But when you actually look at this parasite, it occurs in the Baltic. Salmon occur in the Baltic, and they get along just fine. But what you find is that there's a heritable difference between the Baltic salmon, which is Salmo salar, just like the salmon out here are, in their ability to deal with, resist the parasite. Now this is the Lauren stock, a Norwegian stock. This is a Neva stock from St. Petersburg in Russia. And this is the intensity of the infection that you get following exposure to the parasite. This is increasing time of exposure. What you can see is the Norwegian stock are much more susceptible, and the intensity of the infection much higher than in the Baltic stocks. In fact, most of these fish end up dying, and most of these fish end up surviving. And it's quite interesting. We did a European project, and some people in Denmark studied the nature of the infection, and it appears that this is almost like an AIDS reaction. So basically, the immune system of the Norwegian fish just can't cope with the parasite, and it goes into overdrive. And here's another one. Same type of experiment that Andy's talking about. We synchronously spawned eggs from two stocks on the Kyla-Sutherland system, placed them back into the rivers. We've also put them into a third location, the Loth, where there's no salmon, and we kept them in the hatchery. We looked at the egg mortality rates for the shin and oikle stocks in these different locations. If you put them in this third location, 99% survival. Remember, you're physically playing around with these things just after you fertilize. That's a pretty good survival rate, 99%. You put them into the shin, you get slightly higher survival rates. Maybe we didn't put them in as well. You put them into the hatchery where you're looking after your eggs. They do quite well, no problem. You put them into the oikle, exactly the same batches of eggs. Here you get mortality rates of about 7% of the oikle stock in the oikle system, and you get 37% shin fish dying in the oikle system. So you get kind of an asymmetrical result. Why are shin fish doing so poorly here, but they're doing well in all other situations? As it turns out, this is associated with acid flushes coming down the oikle system. And the acidity actually affects the egg membranes and causes them to harden and makes it difficult for the fish to actually get out of their eggs. And when you look at the mortality, that's exactly what's happening here, but the oikle stock is capable of actually dealing with that in a better way than the shin stock is. Yet these are two tributary rivers of the same river system. So this is variation within a river that you're dealing with here.